Now I would like to invite Georgia Olukaidou Gogo from Psycho in order to give the awards to Dr. Elpiniki Ninou from Greece, whose work she's investigating microglial signatures in models of familial Parkinson's disease, aiming to elucidate the role of microglial cells in neurodegenerative diseases. Congratulations.
Thank you.
Thank you.
Thank you. This is a very nice award, actually, very signing. Thank you. So I'm Elpiniki. I'm a neuroscientist, and my main research focus is on microglia and Parkinson's disease. Okay. So first, what is Parkinson's? It is a neurodegenerative disease mainly affecting the central nervous system. We usually have in mind that Parkinson's disease patients suffer from tremor and other motor symptoms, but the truth is that actually what they experience is much more complicated. So the Parkinson's causes are not fully understood yet. We know that some cases are familial, meaning that the cause is an inherited mutation in a gene, and we know several such genes. But other factors, environmental factors, are under investigation as well, mainly toxins and agricultural chemicals. So in any case, the pathology starts in brain structure called the striatum, and more specifically in substantia nigra pars compacta, what a name, where in Parkinson's patients, this is the region where the dopaminergic neurons start to degenerate. But what causes these neuron loss? Well, the hallmark of the disease is the formation of these Lewy bodies. These are aggregations of proteins and mainly alpha-synuclein in the neurons, causing subsequent axonal degeneration. So actually, this neuron loss can be the cause, can be caused by impaired autophagy, mitochondrial dysfunction, or the accumulation of misfolded alpha-synuclein, and of course, neuroinflammation. And here is where the microglia cells enter this difficult equation. So the microglia cells are, as I like to call them, the immigrants. They are immigrant immune cells in the brain. They enter and colonize the brain during embryonic development. They are derived from the yolk sac, and they stay there forever, serving as the guardians of the brain. In Parkinson's disease, these cells, the microglia, actually recognize and internalize alpha-synuclein. But then, due to impaired clearance, these proteins are spread to neighboring cells. These cause microglia activation and leads to persistent neuroinflammation and disease progression. So our current projects focus on the very early microglia changes in an in vivo mouse model of the disease, also to changes in microglia after maternal immune activation and the sex-specific dysfunctions in microglia in Parkinson's disease. Of course, you'll know that science is a teamwork. I'm very fortunate to be part of a great group in Hellenic Pasteur Institute in Athens. And I would like to draw your attention to this girl here, CJ Karad. She joined our lab last year, and together we founded actually our collective, In Vivo Nervous Tales. So this collective explores, let's say, the link between art, science, and human experience. You can find our science communication videos on Instagram and other fora. And we are very happily and proudly just starting to form our biggest project, which is BioArt, the Mind Unraveled. It is actually an exhibition blending neuroscience and art, inviting audiences to reflect on how science and society shape our perception of truth and reality. We seek funding for that. Thank you once again. Thank you.